Okay, so announcements. The um, the reading assignment. I forgot my good pointer at home, so I have to I'm stuck with this old laser pointer. But the um, the reading assignment and the Canvas assignment are due Sunday. The Canvas assignment is now available. Since I didn't make it available till today, I have the availability window until Tuesday night. So basically until Wednesday morning. So you have a full week to do that Canvas assignment. It covers everything in Unit C, which will be stuff in this lecture as well as stuff on Thursday's lecture. But there's no time limit, so you can feel free to start the problems and then the stuff at the end that if you feel like that isn't clear from reading that you just want to wait till Thursday's lecture, you can do that Thursday. Um, I also put the reading exercise and perusal for Unit D, which is about communication. I actually think that's a pretty cool reading about these different communication modalities in crickets and how they choose when to chirp and when to trill versus when to just walk around and look for mates, and how that depends on density and some other things like that, and the energetics of all of those things combined and how it kind of comes together. So I think not only is it a good communication article, but you'll see stuff about heritability and some other things in there as well. So definitely, I'm excited for that article. So that's out there, but that's not due until the end of Unit D. And then the timeline going forward. So here's us today, lecture C2. Then we've got cognition on Thursday. And then we'll start communication next week, 3 lectures on communication. So this will be Tuesday of next week, Thursday of next week, Tuesday of the week after that, and then a midterm review. So that will just be kind of highlights and questions. That will be that Thursday of that week. And then we'll have the week after that, stage 1 and stage 2 of the midterm. So I'll probably have this on all the reminder slides from here on out. So we'll gradually count down so we can sort of feel that midterm coming up sooner there. So as I've mentioned before, at the top of every unit, there are study guides that have sort of the things that you should know coming out of the unit. So knowing, being able to assess whether you know those things is a good idea. To have terms that are listed. So those are the terms that I'm hoping you should know. It has links to the lectures, but I've also set up little highlight slides, which will be what I present if there are no questions on the midterm review, where I've gone through every one of the lectures and just grabbed the slides that I think are sort of the most important or address like the key concepts, so that you've got kind of a greatest hits of all those slides in one place. And so those are again in that study guides for each one of the units. So if you go to the modules on Canvas, you open up each unit, the very top, you'll see like Unit A study guide, Unit B study guide, and so on. So those are the things that I would recommend you kind of glance over and sort of see, do you feel prepared for that midterm coming up? So that's kind of our timeline going forward. Any questions about this? Yeah, that's here. So I don't have the dates off, but so this is today. This is Thursday. This is next week. This is the week after that, and this is the week after that. So this is, it's so if this is one, so if we go ahead 1 week, 2 weeks, it's basically 3 weeks away. And remember, this is a stage 1, stage 2. So on Tuesday, you can come into class to take it. It will be a Canvas-based exam, or you can take it any time that day. But whenever you start it, you'll get 90 minutes. So if you come to class, you might be restricted to 75 minutes, although it doesn't seem like anybody comes in after class. So I can probably fudge it a little bit on the end there if you did want to take it in class, but you don't have to take it in class. So that's individual based. You get a sheet, front and back of notes. And then on Thursday, it's the exact same exam again. You can come to class again if you want, but you don't have to. I will be here both of those days at this time. But then you can work together with anyone else in the class and with your notes, any of your class materials, and you'll get 80% of your exam score from stage 1 and 20% of your exam score from stage 2. So just as a reminder. Any questions? Yeah. I did have it on the calendar there, and if you look at the tentative there's like right on the front page, then also up at the very first module, the course information page, there's a tentative activities schedule. If you click on that, it will say when these are. So it's on the calendar, the Canvas calendar. It's also in that little schedule there. And so that's to get and as we get closer, I can send announcements out too, but it's on Canvas right now. Look for that tentative schedule and look for your Canvas calendar and go ahead to probably like first week in October. That sounds about right for these. Okay. Any other questions? All right. So this pointer is a little more problematic than there we go. So we didn't really cover associative learning, so I'm going to basically make sure that we cover that well this time. But last time, the goal was to cover all three of these things: imprinting, habituation, and sensitization. So non-associative learning and associative learning. So just as a review of that, there we go. So imprinting is this kind of famous experiment with Konrad Lorenz, where if you gave a quack during a critical period, so you give a critical, we'll say, signal during a critical period of development, and he got these ducklings to follow him as if he was their parent. And so that's an example of imprinting. So during a critical period, there is a learning of a particularly informative and important cue, where that cue is assumed to be salient and consistent for the rest of your life, but it might be a different cue than others. So your parents are, for the development up through maturation, they have the same parents, so they only have to remember one parent. So there's a critical time they remember which parent, but then once they figure out which parent, then they don't have to keep relearning that. So they always know which duck to go to. And in this case, the duck was Lorenz. And so that's imprinting, recently popularized by this upcoming movie, which has a very similar story where a robot is imprinted on or a duck imprints on a robot, and that robot then takes care of this duck, and hilarity ensues. So, so that's, in popular culture, the exact same thing. So now the other thing is, any questions about imprinting? Sort of one of the most basic forms of learning. Yes. Well, the conceptual model in imprinting is this idea that it is an essentially permanent thing, that is there's a critical period in which is a window of time that you learn that thing, and that there isn't a normal way that you can sort of break that. Now the actual mechanisms that go on here, that doesn't mean that there's absolutely no way that there's that you can forget this or anything, but the critical feature here is that this is a type of learning where you're most plastic during a critical period of development when you're sort of given the right password. So in that case, in Lorenz, he couldn't just show up and look at the ducks. He had to like quack at a particular time in their development in order for them to say, that's got to be my parent. It made the quack right at the right time. So that's the idea here, is that the plasticity peaks during a critical period of development, when there usually is some critical association that once you see that, okay, yeah, that's the quack that I was listening for, then that becomes the learned association. Okay. So then habituation and sensitization, these are non-associative learning processes where it acts as an informational filter. So as you're going through life, you receive a bunch of different stimuli. And so the idea here is that not all of them are informative. And if you had to attend to all of them, it would be very difficult to be able to queue up the right responses. And so those stimuli that are relatively low intensity and repeated over and over and over again and don't seem to have any associative consequences to them, then your central nervous system, your brain, starts to learn to ignore them. And so it stops responding to them. So there's an initial response, and then it stops responding to them. There is a very similar process that happens in your peripheral nervous system. So out at the tips of your fingers, called sensory adaptation. And in that case, if you have a constant stimulus, then your nerves actually will stop sending signals to your brain about that constant stimulus. So this is happening at two different levels. You've got habituation going on in your brain for the repetitive signals that happen to hit your brain because there's no memory in order to figure out that things are repeating at the level of your at the level of your nerves, at the level of your peripheral nervous system, out at the end of your nervous system. And so all they can reject are these constant stimuli. So that's what sensory adaptation is. But when there's this periodic thing like a hum that that it keeps asserting itself and it's not, too obnoxious, but it's loud enough for you to hear that over time your brain, it keeps getting those signals from your peripheral nervous system and your central nervous system stops paying attention to them. And that's habituation. But then, if one of those things that have been habituated to asserts a really large stimulus, then you can get sensitized to it. And so there are some types of things where, if you hear them, you start to ignore them, but there's just certain things that if they're just loud enough, it's like your brain fixates on them and it can't help but hear this like clicking noise or something like that. And maybe it's just random enough or something like that. And it just, for whatever reason, you become sensitized to it. And so you actually have become hyper attentive to that thing for good or for bad. So in principle, these two things together form an informational filter, which make it so that it helps you filter out things that aren't informative, but at the instant they become super informative again, or potentially informative, makes you hyper aware of them. So it's supposed to be a filter, an informational filter, the combination of these two. But you can have them be maladaptive as well, and that can lead to you being hyperfixated on something that maybe you don't need to be hyperfixated on. So that's habituation and sensitization. It's non-associative learning, but it is a change in your central nervous system. So we still refer to it as learning, and it's still a plastic deformation of the stuff that goes on in your brain, but it's not necessarily being associated with one thing with another, like the associative learning we're about to flip to. So any questions about habituation or sensitization? Yeah. So an associative learning is you actually associate a a particular stimulus with a particular response. So you'll say, "Oh, whenever I see red, I turn my head," or something like that. Non-associative learning here, this is it's not you're not associating the particular stimuli with the behavior. In which case, you're just learning to ignore the stimuli or attend to the stimuli. So it's a more generic learning. Yeah. You said, is there like a field that studies these? I mean, I wouldn't necessarily say there's a specific field. I mean, I would say that these are well-known fundamental learning processes that whenever we talk about any sort of complex learning process, the question is always like, will the animal or the human habituate to this or not? And so it's just sort of one of the things we look for. So, for example, slime mold, which do not have neurons, there has been studies that show that if you they don't like light. So if you shine a bright light in the middle of a slime mold, which is basically one giant cell the size of a dinner plate. It has multiple nuclei. It's really big. It kind of looks like dog vomit. So up in the where I grew up in Ohio, there was you'd go outside and there'd be mulch outside, and you'd see this plate of what would look like dog vomit, and later in the day it would just be gone because it moves. And so it's this sort of organism that is a single cell, but a giant single cell. and it doesn't like light. So if you shine light on it, it tends to move away from the light. And so people have done things like, what happens when I pulse light and so on. And if you pulse light above it, you get these pulse responses. Whenever the light's on, it recedes away. When the light's off, it comes back. And if you pulse light at the right frequency, you can actually get the slime mold to habituate to the light. And so that's sort of an example of the slime mold learning. Now, it's not associative learning, so it's not the type of learning that everybody's really excited about, but it still shows that there is some memory in the slime mold that somehow knows that whenever you get these frequent, reinforced, low intensity pulses of light, that they're actually somehow not harmful and it's not beneficial to respond to them, and it just stops responding to them. And nobody knows the mechanism for how it's doing the habituation. But those particular scientists, that was a project done by Audrey Dussator and in France. And so she looked for habituation because habituation is just kind of a foundational topic in learning. So it's just one of those things, whenever we're talking about learning, we say, well, what type of learning are we looking for here? And then we know how to assess it in different ways. Dussator, Dussutour. She's sort of very well known as sort of the modern kind of expert in all things slime mold. And that particular organism, and if someone's interested, I could email you, but the genus is Physarum, and the species of the specific name is Polycephalum. So Physarum polycephalum. It's sort of this orange. It's a myxomycete protist. So it's a multinucleate protist. So it's a very weird organism. All right. Any other questions about habituation or sensitization? Okay. So, assuming this. All right. And then the big ones, associative learning. So, otherwise known as conditioning. So, in conditioning, this is a form of learning where we make an association between a stimuli and an action or a consequence of that action, a response. That's the big difference. In the non-associative learning, there's no response. It's just ignoring the stimulus or paying attention to the stimulus. Here, you're actually coupling it with a behavior. And so there are two major schools of thought when it comes to associative learning. There's operant conditioning and classical conditioning. Classical kind of came first with Pavlov's dog. And then Skinner came in and said that well, really, that he thought that this wasn't a very natural way to study behavior, and he introduced operant conditioning to sort of match more what was in kind of nature. So what is going on with these two types here? So in classical conditioning, this idea, so if you've heard of Pavlov's dog, this is actually a figure I think that Pavlov had in one of his early papers of a setup where a dog is kind of harnessed and has a food bowl, and there's different stimuli that they can give. And the idea here is that you're associating a relevant natural stimulus that already exists in the repertoire of the animal and already has a response, and you're associating it with a new stimulus that the animal does not currently have that response for, or perhaps any response for. So it's an idea that you're transferring or copying a behavioral response from one stimulus to another. So we start out kind of in this case right here. We have the so-called unconditioned stimulus, the smell of the food, and then the original natural response, the salivary response of the mouth preparing to receive the food. So that's just already in the animal at the moment. And so we then have a neutral stimulus. So you ring the bell. Well, right now, if you ring the bell, the dog doesn't do anything. It hears the stimulus, but it doesn't have a response to ringing the bell. So at this moment, we call it the neutral stimulus. And so when it gets the neutral stimulus, nothing happens. So what you do in classical conditioning is you pair a stimulus that the animal responds to with a stimulus that the animal does not respond to. And by doing so, if you do that over and over again, then you can form a new association between the previously neutral stimulus and the behavior, the response that you used to only get with the natural stimulus. So I didn't create a new response. So it's the same salivating, dog. It's the same drooling dog is the same response here. But now it drools when it hears a bell or smells food. Before, it only drooled if it smelled food. So now we've copied the drooling response from one stimulus to another. That's classical conditioning. In the honeybee example that we talked about last time, the honeybee sticks out its tongue, and it naturally sticks out its tongue with certain odors and sugar. So you can cover its antenna in sugar water, and it will then stick out its tongue as if it's walking around in a flower and it's gotten nectar on its antenna. That it's like, oh, I'm going to stick out my tongue and I'm going to drink some of that nectar. Well, if you then pair the sugar water on its antenna with an odor that it also can smell on the antenna, then over time, if you just give it the odor, it will stick out its tongue. You don't need to pair it with that sugar water anymore. And that's the same thing going on here. So that's what you should remember with classical conditioning. Classical conditioning about transferring, copying, adding a stimulus to an existing response. So any questions about that? Okay. All right. So that's classical. Skinner wasn't impressed with classical conditioning and thought that they could do a lot more with what he called operant conditioning. So this is a term that Skinner introduced, I believe, operant. An operant is a behavior that results in a consequence that can be learned. That's very generic, but basically what Skinner was saying is that you can have an animal do a just sort of behaving as it does, doing, bumping into things, running around, checking things out. And at any particular time, an animal does a behavior that you like or dislike, you can reinforce it or punish it. So the idea here is changing the frequency of the behavior. So the animal would normally do this behavior, once, one in every, for 30 seconds every 5 minutes, and you would like the animal to be doing the behavior much more often. Well, then you want to reinforce it. Or you don't want the animal to be doing that behavior, and so you punish it. And so reinforcement is a term we use for anything that leads to an increase in the frequency of a behavior. And punishment is a term we use for anything that leads to a decrease in the frequency of the behavior. And there are two ways that we can do reinforcement or punishment, and then I'll pause for questions here. Positive and negative stimuli. So we can apply stimuli, or we can take stimuli away. And those stimuli can either be, you can think of them as reinforcing stimuli or punishing. I wouldn't say them that way. You can think of them, those stimuli, as either being rewarding or being averse, being painful, or so the idea here. Let's say I do, like, for example, positive reinforcement. What does that mean? Well, that means whenever an animal does something that I want them to do more, I give them a reward. I've added a stimulus. So adding a stimulus means positive. To increase the behavior, that's reinforcement. Now, what if that animal instead did something that I didn't want them to do? Well, in that case, if I add a stimulus that they don't like, that's positive punishment. So I've added a stimulus to decrease the frequency of a reward, positive punishment. Now let's go the other way. Now let's say I've been feeding the animal, nice, tasty sugar water continuously, and then it does something that I don't want it to do more of. I might take the sugar water away when it does the thing that I want it to decrease. That's referred to as negative punishment. It's negative because I've removed a rewarding stimulus in an effort to decrease the frequency of the behavior. And then finally, negative reinforcement. Let's say I've actually applied, the animal is constantly being baked in salt water or something like that it doesn't like. It doesn't like the taste of salt water or whatever. And at the instant it does a behavior that I want it to do more of, I stop, feeding it the thing that it doesn't like. That would be negative reinforcement. I've taken a bad stimulus away in order to increase the likelihood of the animal doing a behavior that I just saw it do. So you can combine both of these with both of those. So there's 4 different approaches to operant conditioning. One of the dimensions is whether you're increasing or decreasing the frequency of the behavior. The other dimension is whether you're adding a stimulus when the behavior happens or you're taking the stimulus away. Questions about that? Yeah. So would you call that what type of positive reinforcement? Right. Adding chocolate to get more behavior, positive reinforcement. Right. Yeah, thank you. That's good. Any other questions about this? Sometimes this gets a little confusing. Negative punishment, negative reinforcement. Negative reinforcement sounds really weird, right? But you just have to sort of separate in your head. What am I trying to do to the frequency of the behavior? And am I adding or subtracting something to make that happen? Okay. All right. So this is often how these things are done in the laboratory. So kind of hidden behind this box here, there's a cartoon of a pigeon in a Skinner box, but they also do with mice. So this is a very general purpose Skinner box right here, operant conditioning box. So it's got food that it can distribute. It's got lights that the animal can see. It's got a loudspeaker that the animal can hear. It's got a lever that the animal can press. It can jump up and press on that lever. It has an electrified grid here. So in fact, I think, did I have a picture of the mouse pressing one of these levers on the slide, the previous slide? Let me go back and see. Yeah, so there's an example of, I think, a rat pushing on one of these levers. So that's the type of thing you'd see in the lab. And so you've got one of these boxes that's got all of this cool stuff in it. So you can apply punishment, negative stimulus, you can apply positive stimulus, you can get behaviors out, you can apply, you can do all, there's all sorts of different conditioning you can do in this very general purpose box. And that's the so-called Skinner box. All right. So the other terms that I want you to know is have to do with whether the stimuli that you're applying is a stimuli that is sort of within the kind of what the animal would normally expect in its kind of day to day. And so that we refer to as an animal can either be prepared if the task you're asking it for is a task that it already knows how to do, and it frequently does that. It's already maybe familiar with that stimulus, and it already knows how to search for seeds or whatever. It can be unprepared if the task has no relevance. So you're trying to teach it to do your taxes or something like that. It's something that it was never in its evolutionary history had to do, and you're trying to train it to do that thing. So there is a bumblebee researcher named Lars Chitka who's trained these bumblebees to do things like pull strings in order to get discs out where they can drink nectar from. And then it's also they train them to like roll little soccer balls around and get the soccer balls up on top of little goals, and then they get rewards. Those are things that those bumblebees have never had to do before. So they were totally unprepared for that. And yet he can manage to train them to do those things. And then there's contra-prepared, which is a little bit like extinction behavior we were talking about before, where in this case, you're training them to do something that goes against what they've been doing in their evolutionary history. So they normally respond one way, and you're training them to go the other way. And so, this, of course, can, depending on what category you're in, can affect how quickly you can get the animal to be trained, or even whether it's even feasible to train the animal that way. So these are sort of the big operant conditioning dictionary that I want you to have down. Is that operant conditioning? That's the one where you're not copying a stimulus. you're adding sort of, you're changing the frequency of a behavior. And you can do that either through reinforcement or punishment, and either by adding stimulus, positive, or taking stimulus away, negative. And it can be in the context of an animal that is either prepared for what you're asking them to do, unprepared, or contra prepared. That's kind of the lingo for what we need for operant conditioning. Any questions about that? Yeah. Yeah, all four of them are possible. So you can have positive reinforcement and you can have positive punishment. You can have negative reinforcement or you can have negative punishment. So I guess I can put parentheses around these. So positive, regardless of what positive is modifying, it means I've added a stimulus. Now, it might mean I've added a bad stimulus, or it might mean I've added a good stimulus. So positive reinforcement is I add something good, positive punishment is I've added something bad. Any other questions? Okay. I'm definitely bringing that other clicker the next time. Okay. So with that in mind, this was the question that I meant to ask you at the end of last time, but I think it fell a little flat. So let's try it again here. So there we go. We've opened it up. So you may not, I've only done it once in this one, but in the last lecture, before every iClicker, I said, we're about to do an iClicker. And at the same time, in that slide, before every iClicker, I put a little star in the upper corner up here. And so if you notice that, and if you saw the star and you started to get out your iClicker and said, oh, the star's coming up, he's about to give me an iClicker, I should get ready for the iClicker, you have been classically conditioned. So in that case, what do we call the star? Is the star the unconditioned stimulus, the unconditioned response, the conditioned stimulus, or the conditioned response? That's the question. I'll give you a second to think about that. So just to set that up again, I normally say, hey, we're about to do an iClicker on the next slide. And then we do an iClicker on the next slide. But behind me, as I'm saying that, there's a star over my head that I have not pointed out to you, but I've just shown that there just happens to be. Whenever I say that, there happens to be a star over my head. So what does the star? Is it the US, the UR, the CS, or the CR? Let's see how we're doing. All right, the results are looking much better than they did at the end of yesterday or Thursday when I had to rush through this. And so I'm hoping that means I've done something better here. So right now we're getting a big peak on one of the answers, and then a little bit of a side peak on one of the others. And so I think we've got, I'll give you 15 more seconds and then I'll hit stop. So get your final answers in. Maybe 7 more seconds. 3, 2, 1. All right. So we got 7% for A, 4% for B, 79% C, and 4% D. And we got one person who did E, which which is fine. And that's the majority of classes. What I was looking for, it's conditioned stimulus here. So I understand why unconditioned stimulus, why we had a side lobe there, because it's obviously a stimulus, right? But it's tricky here, the unconditioned versus conditioned. So the idea here is that your natural behavior is to listen to me say, we have an iClicker, and get your iClicker out. That's what makes me say, hey, we have an iClicker. That's the unconditioned stimulus. Whenever you hear me say, we have an iClicker, you respond by getting the iClicker app out. That's the unconditioned stimulus. The natural stimulus that leads to the natural behavior. It's totally unnatural for you to see a star and think, I should get my eye clicker out. There's no reason that that should be your natural response. But you've learned that whenever I say we're about to do an eye clicker, there's a star over my head. So that must mean that the star also means that we have an eye clicker coming up. That's what makes it the conditioned stimulus. So it's a new stimulus that has gained meaning through association with the existing stimulus that already had meaning. So any questions about that? Does everybody feel comfortable now that the star in that example is the conditioned stimulus? Okay. And then the response being you get the eye clicker out. So that the unconditioned response and the conditioned response are actually the same response. But the idea here is we call it the conditioned response because we're saying, like, now that we have a conditioned stimulus, what do you do with the conditioned stimulus? Well, you do the conditioned response. And then if you have to think, well, what was the conditioned response? It's the thing that was transferred from the unconditioned response. So it's like we're copying a response from the unconditioned pair on to the conditioned pair. That is classical conditioning, copying from one thing to another. Everybody feel okay about that? All right. All right, so new stuff. Learning in the wild. So how do we use this stuff to think about more complex learning examples? So I'm going to use dog training and Dexter and his little lion outfit as an example. So if we look at dog training here, so there's me showing Dexter a little sign that says sit, and then me showing Dexter a sign that says down, and a second, if he's a good boy, which of course he is, lays down. Dog training is a really cool example because it is this combination of operant and classical conditioning. And what do I mean by that? So, step one, operant conditioning, get the dog to do the thing you want them to do more often than they automatically do it. So at this point, you're just trying to make the dog do something more frequently. So you typically are just reinforcing a behavior. Like, a dog usually doesn't want to bring their paw up into the air, but if I want them to wave, that's what I want. So if I happen to get the dog just to do a little bit of that, I then reinforce that behavior. And then the dog just starts doing that behavior more and more. And until eventually they're just, waving at anything. They don't know why they're waving, but they know that whenever they wave, they got that reward. So that's step one, is just getting that behavior, creating the unconditioned response, creating that in a certain context, the dog is just going to start doing that. And then you take that and then it becomes classical. So now we can add things to that. So whatever I was doing to get the dog to wave, now I want them to wave when I say the word wave. So I now get them to copy whatever stimulus made them wave before onto other stimuli. And so if you ever do training like this is there's variations to this, but roughly speaking, this is what you do. Is like people will always ask when they start training animals like this, when do I add the word in? Because everybody wants the word, and a trainer will just say, don't worry about the word right now. We just need to communicate to the dog what we want them to do. And that just means reinforcing some behavior, just getting them to do it more frequently. And then once they do it more frequently, then we tune it by saying, okay, actually, that thing that you've been doing, let's actually only do it when you have this stimulus. And then, once you've got this stimulus, then let's only do it when you hear this other, or both of these stimuli. So you can take that behavior and copy it to all sorts of other stimuli. Once you've trained them once, you can then just copy it to other things. And that's classical. So classical is copying, and then, operant is original, O for original, C for copy. So any questions about that? This is not a perfect analogy. It's not a perfect model. And that's kind of important because as we'll see through the rest of the examples here, the basic models of learning are not meant to sort of perfectly fit everything here. So if you're like, I don't know if this is completely operant here, that's totally fine. But I hope you sort of see that operant provides us a way of thinking about this first step, and classical provides us a way of thinking about this second step. That's the important thing I'm going to motivate here. So, with that in mind, what we're basically going to do is we're going to take these three classic models here, and we're going to look at behaviors of more complex learning in nature and see what these models tell us. And sometimes these models give us interesting ways of thinking about the behavior, like in the slime mold case, like, oh, that's habituation. Other times when the models don't fit, that ends up highlighting new causal questions that we might not have thought about and maybe showing us mechanisms that we didn't think existed. And so, as an example of that, so this is called taste aversion learning. So this is widespread across animals, is that if a lot of animals, if they eat something like a poisonous butterfly, and after they eat it, it causes them to get sick and empty the contents of their gut. That's pretty costly. It may make them, unwilling to eat for a while, and it's a really, it's not a pleasant thing to go through. And so, what they'll do is they'll learn then that after this one encounter, to never eat that butterfly again, never eat a butterfly that has that color pattern again. That's what we refer to as taste aversion learning. Now, when you first hear about that, I just want to highlight in as little as one strongly negative reaction, an animal will learn to avoid that taste for possibly its entire life. One bad, one extra, one bad sangria at the bar, one bad butterfly, one bad mealworm, and suddenly for the rest of your life, you don't want that ever again. That's taste aversion learning. It is something special because if you think about it, it's not quite associative learning. It's way, way too fast for associative learning. We'd like to think you're associating that butterfly with that's a reduction in the frequency of wanting to eat the butterflies. But you've seen those learning curves. It's very rare for a learning curve to pop up to 100% learning after a single trial. It's just too fast for associative learning. And also, because it's for the rest of your life, it doesn't quite feel like imprinting because it's not like it's happening at a critical time in development. And so, to add even extra information here, this learning can happen even when an animal's under anesthesia. So you can knock an animal out, feed them while they're knocked out, something that will make them sick, have their body, they're physiologically, get sick while they're knocked out, and then their brain still remembers that association and will avoid that stimuli later. So it's like the taste, so it's not necessarily, remembering the color, but it might be that immediately once you taste that butterfly, you spit it out. You don't even swallowing it. And then on top of that, I want to emphasize here that there can be 24 hours between when it's eaten and when this actually happens. That's also, in associative learning, you've got to follow like that stimulus and like if you want to reinforce or punish a behavior, that reinforcement or punishment's got to come right then. Like if it's too many seconds later, it's not going to be properly associated. But an animal will somehow remember that 24 hours ago, it ate this thing and it caused it to throw up. And that, led it to then say, I'm never, putting that, like if I taste that again, I'm immediately spitting it out from then on out. So, and then on top of that, what's really interesting is there are some animals that do not do taste aversion learning. So vampire bats, for example, vampire bats are, there are, microbats, macrobats, they're bats that eat insects, that are bats that eat fruit, and then there are bats that drink blood from mammals. And the other bats, fruit bats, bats that eat insects, they have taste aversion learning. All of the bats, whether they're vampire bats or not, can do associative learning in the laboratory. But they do not do taste aversion learning. So regardless of how sick you make the bat, it will not learn that thing that it tasted that made it sick is something it should avoid later. They just do not have the ability to do that. And so, you might ask, well, why would this might be the case? And if you think about it, like emptying your gut is really costly, especially if you can't eat them for another day or whatever. So you really should only do this like emptying your gut when you're sick, if it's really important for you to get rid of these poisons. And if you think about vampire bats, it's really rare for them to drink kind of toxic blood, right? Like, spoiled fruit, that's one thing. An insect that, a dead insect that has, some parasite in it or whatever, that's another thing. But, blood is a pretty clean thing to eat, relatively speaking. And so, the vampire bat really doesn't have a reason to have taste aversion learning. And apparently it doesn't. So, rather than risking throwing up for no good reason, it just does not have that circuit. And I say circuit because the best evidence that we have is that taste aversion learning is actually a totally different neurological process in the brain than associative learning. It's like a totally different circuit. And so you might have the circuit or you might not have the circuit. So vampire bats don't have the circuit. So it feels like associative learning, but it's a totally different type of learning, the same way imprinting is a totally different type of learning. So this is one of these cases where we have these basic learning models and we use them as kind of a lens on which to look at a behavior. And if it just doesn't quite fit all of them, it motivates us to ask more questions, to test new hypotheses, and lead to sort of very interesting results here that taste aversion learning is something special. All right, so any questions about that? Taste aversion learning, what I mean by it being sort of different. It's learning, it's plasticity, but we wouldn't call it formally anything like associative learning or imprinting. It just doesn't fit into the basic categories. Okay. Okay. Now, there are some things that fit perfectly. So this is a Steller's Jay. It's a beautiful little blue bird. Shows up on campgrounds. You can find it up in Flagstaff. Um, a lot of people think they're really annoying because of how smart they are. So you can put things on your campground that keep birds out, and the Steller's Jay will just keep trying all sorts of different things until it manages to break down your mechanism and get in. They're extremely clever, and so they're really well known for their use of so-called trial and error learning. They just try and try and try. They keep trying random things over and over again, and eventually they find something that works, and then they learn how to do that, and then they'll keep doing that over and over and over again because it gave them the reward. So what type of learning does that sound most like, trial and error learning? Operant or classical? Let's think, are we changing the frequency of a behavior or are we transferring a behavior from one stimulus to another? Operant, right. So we're increasing the frequency of this weird behavior that the Steller Jay found on its own. So it's like it's training itself. It's doing an operant conditioning experiment on its own where it knows there's a reward and just keeps trying things that finds that reward. And so this fits the operant model perfectly. So this is an example where our lenses, our basic models, help us make sense of what's going on when this Steller Jay is freaking out at this mechanism and eventually pops the mechanism open and gets, the peanuts out or whatever out of it. It's also really good at getting into nests of other birds and destroying their eggs. So, it's a voracious forager that will use its wits to get anywhere it needs to be to find food. That's the Steller's Jay. So it's a cool little bird there. All right. So that's kind of a model that fits really well, but we can kind of then go on to more complex behaviors and think about what these learning models can help us. So I show up this squirrel here, this tree squirrel here, as an example, as an opening to this hoarding behavior that in the reading for this unit, it's all about hoarding and cache retrieval. So that's kind of what we want to talk about here. So It doesn't sound that exciting, hoarding or caching, but turns out that from a learning perspective, there's a lot of really cool stuff you can study there. And I hope those of you who read the article can see that there's a lot of interesting hypotheses that you have to test to really figure out what's going on. But I'm kind of getting ahead of myself. There are two. So basically, an animal can either take down a prey and when it's done eating, leave it there to rot, or it can find as much food as it can find and just store it away. Now, there are two different, and that's what we refer to as caching. And so there are two types of food storing behavior, two types of caching behavior. There's the type that you can, that, honeybees and say harvester ants, messer in this case, do, where you come back and you store them centrally in one location. So you know where your food stores are. They're, in your nest or in all of your sub nests. They're not out there. So you don't have to, it's easy to find because it's just right there where you are. The alternative is what these tree squirrels do, where you scatter hoard it. So this is referred to as larder hoarding. So you're hoarding into a central location. The problem here is that you have to defend against it. So there are, for example, stingless bees there's other bees that will then try to raid the stingless bee colonies in order to get all of the resources that are inside there. In fact, there's a lot of bees that are like that. There's a lot of ants that are like that. So defense is the major behavioral problem, which is another chapter that we'll talk about later. But if you alternatively do this strategy where I'm not going to hide it in one place, I'm going to hide it everywhere, then that's a great, I'll get to your question in a second, that's a great way to not have to worry so much about the defense. But then it introduces a new problem that how do you find it later? And then another problem is that if you're not controlling it, how do you prevent it from being stolen by another animal that happens to find it later? Question? Yeah, larder hoarding is when you're all in one place. Scatter hoarding is when you just randomly, well, I don't want to say randomly, you place it throughout your home range. You don't do it home. Not necessarily everywhere, and that's what we'll get to. But you don't have a home where you put all your food. You put your food in spots in your home range. Where you put them might vary based from species to species, but the point is you're not putting them all right here where you live. All right, questions about that? Clear? So we focus because of the learning emphasis here on scatter hoarding. So scatter hoarding, the big question is how do hoarders retrieve items from their cache? So what type of question? This is a causal question. It starts with how. So would we call this mechanism, phylogeny, ontogeny, or function? Anybody? I hear function back there. I hear mechanism. Any other guesses? How many people would say function? How many people would say mechanism? It's more for mechanism. One of the key things is because it starts with how, we generally know that how questions should put your mind in either the development or the mechanism arena. Those are the how questions. Why questions put yourself in the evolution, phylogeny, or function. So here, we're not hypothesizing on the function of the scatter hoarding. We're just saying, how do they manage to find these items? We don't know if it's the good way to find it. We don't know if it's good to do scatter hoarding in general, but just in general, we know this squirrel hid a nut over there. How's it going to find that nut again? Now, this isn't something about how the squirrel develops. It's about how at this instant of time you find that nut, and that's why I would call it mechanism. All right, so there are several hypotheses that we might want to test, and not every scatter hoarder does it the same way. So for some species that do scatter hoarding, one of these will better fit. For other species, another one would better fit. So, when we do tests, we never can assume. We have to say, for this particular species, how do we discriminate among these options? And these are the three major options that we think about whenever we talk about scatter hoarding. Well, the one idea, the simplest idea to how you find the things that you've stored is just to look for them again. So what if you don't learn at all? What if you just place them haphazardly? And so at this point, if you want to find that nut, you just go searching for nuts again. But maybe because you went far off to find the nuts originally and you just placed them kind of closer to you, there might be, a greater chance that you're going to be able to find them, but you have no sort of ability to bias your search towards one area or another. You haven't learned anything, and you don't have any rule for how you hit it anywhere. That's kind of the simple, almost the null hypothesis, really, that you're just going to forage again for the stuff that you hid. Not a soup doesn't seem like a very smart strategy. The next step up from that is the so-called searching by rule hypothesis. So you can imagine that maybe one thing you do is I only bury the things that I found under certain types of trees. Or maybe I bury them at a particular level that allows me to smell them. Or, so there's this sort of rules that basically means that I don't have to learn where I put them because I'm going to put them in a consistent place that later on I just go to those types of places and it'll be very likely that I find things there. That's what I mean by searching by rule. So it's interesting that like this is on one hand, it's a hypothesis about how you search, but it's really a hypothesis about how you hoard. So it's, you can't search by rule unless you hoarded by rule. So that's the idea there. Now, the alternative to both of these is that you learn. So you say, all right, I buried a nut here. I'm going to look around. I'm going to see everything around me, around where I buried this nut, commit that to memory. And then later on, when I need this nut again, I will go back to those things that I remember. The smells that were in that area, the unique smells that were in that area, the sights that are in that area, et cetera, et cetera. So it's so you're actually making a change in your memory, and that's what differentiates learned cache retrieval from searching by rule. And if you really think about it, it's hard to fit this learning directly into one of those simple boxes, but it's still kind of a version of associative learning and conditioning. It's still this idea that I go into an area and my response in this area, if I'm just standing here, is to dig up the nut. But I'm now associating that response with these new cues that are around me. So I'm sort of transferring my desired response to dig up the nut to this new stimulus of this visual surround. So we still can use, say, classical ideas for how this learning is happening. So it's not totally useless that we have these basic learning models, but it's really hard to sort of shoehorn this entirely into saying, oh, this is definitely a classical conditioning problem, for example. So to clear these different hypotheses, random searching by rule, or learning what's around where you buried things. Okay. Now, the other half of this is once you leave the foraging item somewhere in the forest, it's unprotected. You're hoping that it's cryptic enough that no one else will find it. But now, if you're another animal that is a pilferer, a stealer, a scrounger, then in that case, it's actually of your benefit to try to maximize your opportunities to find the caches of others. So if we can flip then to thinking about the learning behaviors of the pilferers, because you say, well, how could pilferers, maximize their haul from stealing from others? Well, it's kind of the same sort of idea. Well, they could just forage, they could just search randomly and dig anywhere and, or go into every cavity and just hopefully they'll find things that were stored there. That's kind of, again, the null hypothesis. Not really a whole lot of intelligence there. It's hard to imagine that you'd get a lot of return from that strategy. They could do what I'm calling searching by cues. So it might be, and this happens a lot, that there are certain habitats where during dry seasons, you can hide things away and they don't give off any olfactory cues or anything else. But during the moist seasons, you can get fungus growth on the things that you stored. Seeds can start to germinate and so on and so forth. And they can start giving off cues that indicate that there is a cache of food there. And pilferers can pick up on those cues. So they can learn. This is sort of like the flip side of the search by rule, but it's not like they figured out the rule. They're just sort of using these cues. Now, the more advanced thing is you can actually observe the hoarding behavior of another animal, and when that animal is gone, go and dig up their stuff. And this happens. There are birds that sit and wait for other animals to hoard things. In fact, a lot of times they're waiting for other birds, even conspecifics, to hoard things. And then they remember that those birds, stored things there, and then they come back to them later. So they have to learn this. And what's interesting, and this kind of gets into a little bit of ugh, this gets into a little bit of what we'll talk about on Thursday, but um, birds that steal from conspecifics, others of their species, in this way, using observational learning, when they see that there are other birds around them, they tend to have much more elaborate hiding behavior. So if a bird is alone in an area, they'll just sort of, hoard something in a relatively, they won't go very far. They won't dig very deep, and they'll hoard the item there. But when certain birds see that other birds are around that do observational learning, then in those cases, that same species that alone will hoard nearby will then go way far away and try to find an area that has like very few other birds around and then hoard there. So it's interesting. It's a little bit like the birds know what the other birds are thinking because that's what they're thinking. Now, I don't want to say that. That's a very cognitive thing for me to say, but it's almost like that. So it's interesting that there's this G by E, there's this gene by environment, that when you're in a social setting, your hoarding behavior is much more dispersed and diffuse and cryptic. When you're in an isolated setting, your foraging behavior is much more local and obvious. And that's an example of G by E right there. It's the same genes, but in the social environment, it was very different than in the isolated environment. So any questions about this, these differences in pilfering behavior and how they kind of relate to learning? All right, so there's a lot of cool stuff in caching and learning. A lot of people study learning in caching animals, but they're a lot more complicated and sort of exciting examples. And so this is sort of a fun one. These whooping cranes. Whooping cranes are a bird that, in natural populations, they basically get they're born at higher latitude, so like Wisconsin area, that sort of thing, and then they fly down to Florida, and the young whooping cranes will follow an older whooping crane down, and that's how they learn this migration route. Well, and there was an area that, through historical accident, ended up losing all of its adults, and they had this captive bred population that they maintained, and they needed to give these poor birds migratory routes. So what they figured out is that they could dress up in these white outfits, which you can see this guy here, and imprint on these birds. And these giant whooping cranes would follow them around as if they were their parents. And then once they imprinted, you could get into these ultra weight vehicles and fly off, and the cranes would follow them. So you've got these humans in these ultralight vehicles flying from Wisconsin to Florida, stopping pretty frequently to refuel because look at that thing. And then the cranes would stop with them, and then they would just keep making this. And eventually the whooping cranes get down to Florida, they learn their migratory route, and everything would be set, and they could recycle this whole thing. So that's another example of observational learning. It's not pilfering, it's a sort of social learning, this transferring of a route from one individual to another. In this case, cross species. It's kind of artificial, this cross species here, but it's mimicking what would happen in nature if you had the adult whooping cranes there for these juveniles to follow. So I don't know, I think it's pretty cool just to think about this person in this silly little whooping crane outfit flying this ultralight from Wisconsin to Florida, which is not a short distance, and then having this flock of giant birds following him the whole way. It's pretty cool if you think about it. But then a little close to my own home, our heart, is what we had in the opening slide here. These are ants, Temnothorax. So there's a bunch of different temnothorax species, or actually a bunch of different ants that have this particular behavior, and this is referred to as tandem running. So this ant up front here, this leader, she has found a new cavity that she thinks might be a good nest for the existing colony. So she goes back and she then recruits an ant to follow her. Now she has no way to just, to say like, oh, hey, I found a nest over there. It's, she can't do like what honeybees do and say, 5 miles in that direction is a good nest. Go check it out. So what she does instead is she puffs a little chemical into the air back at the nest that we originally saw here, and that brings an ant to her, and that ant will touch her on the rear end. And at that point, she'll walk a couple of steps forward and wait to be touched on the rear end again. Meanwhile, the ant behind her, if you kind of watch, she's looking back and forth. And so we think that she's actually encoding her visual surround, because what will happen is eventually these two ants will get kind of the end of this pas de deux at the final nest here, and then they'll break apart. This ant will go back and she might recruit another. That ant will check out the new nest, and then she will return, and she's able to find her way back. And if she wants, she can lead another one of these so-called tandem runs. What I like about this behavior that it kind of shows here, and we, not I say we, but there are researchers here, actually even at ASU, that have had this tandem running in like a giant area where you kind of print a picture of a laboratory on the inside of a cylinder. And after they go in one direction, you can rotate the cylinder. And then when they come back, they end up missing the nest by the amount you rotated. So it does look like they're encoding the visual landscape as they're kind of going back and forth between these kind of touching here. So what's really interesting to me is like these ants are sort of, it's like a flow control. Like this ant can't communicate a lot of information to this ant. So instead, she just guides her through a series of visual cues. And this ant learns them. And again, sort of related to classical conditioning, kind of getting these new cues that are copying onto this behavior until eventually they find the target nest. And eventually then the leader ant will go back and the follower ant will check it out, and so on and so forth. So this is what we refer to as tandem running. It's another example of sort of observational or social learning, the transfer of information. But I just want to make that key point is in these migratory examples, it's super complex because again, they're maintaining connection while the one in the back is actually getting the information from the world, not from the leader. So it's this weird thing where you don't know what visual cues the follower is picking up, but you want them to pick up some visual cues in order to get them from point A to point B. And that's tandem running. Any questions about tandem running? So these are ants that don't leave pheromone trails. A lot of people think that, all ants do pheromone trails. There are a lot of ants that do. The ants that do them in very different ways. So there's diversity in trail laying. Some people think this is an evolutionary precursor, phylogenetic, hypothesis here. So evolutionary hypothesis. Some people think this is an evolutionary precursor to trail laying, so that eventually ants did get the ability to get trail laying, but this is maybe where it started. That's not necessarily 100% supported, but anyways, it's cool behavior to watch. Okay, questions about that? Yeah. When she got back to the original nest, she puffed a little bit of a smell into the air that said, hey, I'd like to show someone a new nest. And that brought an ant to her. And she may have a little puff of chemicals behind her that maintains connection. So the other ant knows that she's there. But it appears like, because these ants have vision and olfaction, they can smell and see. It appears like they're primarily using their vision to get visual cues and primarily using their smell to maintain connection with the other ant in front of them and the kind of tactile. So the smell doesn't communicate any more information than I'm right in front of you and waiting for you. Touch me when you're ready for me to go the next couple of steps. Okay. All right. So that's kind of like a migratory behavior. So the basic idea here is that we learn these basic models in printing, non-associative learning and associative learning. And they're lenses for looking at complex behaviors in the wild. But complex behavior in the wild are just like that. They're complex. They're not usually going to fit into one of these boxes. So it might be like the dog training. It kind of looks like it's one followed by the other. Or it might be, the extreme case of the taste aversion learning, where it turns out that because it didn't fit into any one of these boxes, it made people research it a little more deeply and found out it's a totally different neural circuit that kind of deserves its own box, its own special box along the lines of imprinting. So they help us find new questions and suggest new answers. They're not meant to be the be all and end all of learning behavior. That said, they do help us design simple, meaningful laboratory experiments that allow us to do learning assays, so that although maybe what a honeybee does in the wild is much more complicated than what she does in the laboratory, we can study differences in how well different bees learn in the laboratory and better understand what are the things, like what are the energetics of learning, how does fatigue affect learning, and so on and so forth. So it allows us to build models that help us understand consequences in complex learning, even if those laboratory models are kind of simple and kind of artificial experiments. And so that's the other thing that we do with them. And learning in real life is complex and will not always have a good fit, but that's okay. All models are wrong, but some are useful. These are just models. They have been very useful so far, but real life does not fit into one of those boxes. All right, so that's kind of the big thing that I wanted to conclude with in this lecture. Next time, we're going to say, well, for really complex behaviors, what are some alternatives? And because it's so easy to talk about that bird that knows how it would steal behavior. So when it sees other birds, it behaves differently. It makes you think that there is a theory of mind that's going on into those birds. And we're going to play with that a little bit. We're going to see where that leads us in cognition. So cognition is an area, or cognitive science is basically a group of scientists that said, I need room for motivation. I need room for things that I can't observe. And that's the only way I can make sense of some behaviors. And that's what we'll talk about to close out this unit before we start talking about communication next week. So with that, any questions about any of this stuff? And there is a clicker. The star was just for the previous lecture. So the only other clicker I have for you is this one. I'll start that up. All right, it should be running. So what type of food caching behavior involves distributing stored food all throughout the home range? Is that larder hoarding, scatter hoarding, central place foraging, or pilfering? And that's all I've got for you. So I'll see you Thursday.